Cancer cells are quick-change artists adapting to their environment. Until now, researchers have assumed that the growth of solid tumors originates from cancer stem cells characterized by specific surface markers, which develop in a fixed, hierarchical order. Accordingly, such cancer stem cells are responsible for tumor progression and produce specific types of more differentiated cancer cells whose fates are predetermined. In a joint interdisciplinary project led by the Luxembourg Institute of Health (LIH), researchers now show that cancer cells of glioblastomas, conspicuously aggressive solid brain tumors, manifest developmental plasticity and their phenotypic characteristics are less constrained than believed. Cancer stem cells, including their progeny, are able to adapt to environmental conditions and undergo reversible transformations into various cell types, thereby altering their surface structures. The results imply that novel therapeutic approaches, which target specific surface structures of cancer stem cells, will be of limited utility. The research team has published its findings in Nature Communications in April 2019. Glioblastomas are the most common malignant brain tumors. Because of their rapid growth, the prognosis for those affected is usually dismal. Many patients hold out hopes for novel therapeutic approaches, which utilize drug-bound antibodies directed against specific markers present on the surface of a subpopulation of immature glioblastoma cells. These antibody-drug conjugates bind to the surface, are then internalized and kill the cancer stem cells. Remarkable cell state transitions. However, results now published in the journal Nature Communications suggest that this approach may be misdirected. We exposed cancer cells in the laboratory to certain stressors, such as drug treatment or oxygen deficiency, explains Dr. Anna Golubievska, junior principal investigator at the NORLUX Neuro-Oncology Laboratory in LIH's Department of Oncology and co-first author of the study. We were able to show that glioblastoma cells react flexibly to such stress factors and simply transform themselves at any time into cell types with a different set of surface markers. This plasticity allows the cells to adapt to their microenvironment and reach a favorable environment-specific heterogeneity that enables them to sustain and grow, and mostly likely to escape also therapeutic attacks. The team of scientists from Luxembourg, Norway and Germany, led by Professor Simone P. Niclot at LIH, proposes that neoplastic cells of other tumor types may be also less constrained by defined hierarchical principles, but rather can adapt their characteristics to the prevailing environmental conditions. The same phenomenon has been observed in breast and skin cancer, says Dr. Golubievska. This observation predicts that cancer therapies specifically directed against cancer stem cell markers may not be successful in patients. The new findings could help to optimize future standard treatments. In laboratory experiments, the researchers were able to show that environmental factors, such as lack of oxygen in combination with signals from the tumor microenvironment, can induce cancer cells to modify their characteristics. This microenvironment, the immediate surrounding of the cancer, comprises cells and molecules that influence the growth of the tumor. Once we understand exactly what causes the plasticity of tumor cells, we can devise combination therapies which target the signals underlying plasticity and thereby improve the therapeutic impact, underlines Dr. Golubievska. Collaboration and funding. The study is a collaborative work between the NORLUX Neuro-Oncology Laboratory and other research units and platforms at LIH. The researchers from LIH also worked in close collaboration with their long-term national partners to whom they are tightly connected through transversal research programs, the Luxembourg Center for Systems Biomedicine at the University of Luxembourg and the Department of Neurosurgery of the Centre Hospitalier de Luxembourg. Moreover, the project was carried out with international partenariats from the Technische Universität Dresden, Germany, the University of Heidelberg, Germany, and the University of Bergen, Norway. This joint undertaking of different research and clinical players gives a truly interdisciplinary dimension to the study. The study is a major part of the PhD thesis of Dr. Anne Dirks, co-first author on the publication, who was supported by an AFR PhD grant number 5,778,172, PhD 2013-1, BM, from the Luxembourg National Research Fund (FNR) and a training grant from the Fondation du Pelican de Millet et Pierre Hippert Faber, Fondation de Luxembourg. Furthermore, the work was supported by funding from LIH, Sächsisches Staatsministerium für Wissenschaft und Kunst SMWK, Deutsche Krebshilfe and Deutsche Forschungsgemeinschaft DFG. Do you hear what I hear? A new study by Columbia University researchers found that infants at high risk for autism were less attuned to differences in speech patterns than low-risk infants. The findings suggest that interventions to improve language skills should begin during infancy for those at high risk for autism. The findings were published in Brain and Language. Humans are born with an astonishing ability to distinguish basic sound units that make up all of the world's languages, says Kristina Denisova, Ph.D., Assistant Professor of Clinical Psychology at the Columbia University Vagelos College of Physicians and Surgeons. But why some infants at high familial risk for autism spectrum disorder are less likely to develop their language-specific competence in toddlerhood has remained a mystery. Previously, Denisova showed that high-risk infants, those who had a sibling with autism, were less likely to turn their heads in response to spoken language than typically developing infants. Denisova says that our team dissociated between head movements in infants at high versus low familial risk for developing autism and detected the signal of future atypical development as early as 1 to 2 months after birth. A large body of research suggests that as an infant grows, future language development depends in part on the ability to distinguish sounds and elements of speech that are familiar versus those that are novel, including elements of pronunciation, such as stress patterns on different syllables. Sensitivity to specific stress patterns in one's language serve as important cues for learning language. In the current study, the researchers studied 52 9 to 10-month-old infants who heard speech with familiar and unfamiliar stress patterns while undergoing MRI. Half of the infants were at high risk of autism. They recorded the head movements throughout the scan and studied whether features of head movements differ between the two cohorts. Low-risk infants turned their heads more frequently while listening to speech with different syllabic patterns, while the high-risk infants did not. High-risk infants had significantly worse receptive language scores and the most atypical head-turning patterns on this task. Those 9 to 10-month-olds who had more abnormal head-turning behavior during 3 types of exposure, listening to alternating stress speech, listening to language, and during sleeping, were more likely to develop autism spectrum disorder by age 3. Denisova studied additional data sets in an attempt to explain what mechanisms might explain the difference in response. Her examination of studies of 774 infants confirmed that high-risk infants have lower receptive language scores compared to low-risk infants, further suggesting atypical processing of speech in the high-risk group. A final query compared vocal learning genes with convergent expression in humans and songbirds, zebra finches, in an important motor region for vocal learning circuitry, laryngeal motor cortex, LMC, against likely gene-disrupting mutations in autism genetics publications. This analysis revealed a potential candidate mechanism for disrupted vocal learning in humans, such that it may be less acquisitive. These findings regarding differences in early language processing in children at high familial risk for autism are intriguing and deserve replication and extension. Of great interest will be whether and how these early differences in language processing relate to the differences in social communication that are so key to understanding autism, says Agnes H. Whitaker, M.D., clinical professor of psychiatry at the Columbia University Vagelos College of Physicians and Surgeons. The study is titled, Failure to Attune to Language Predicts Autism in High-Risk Infants. Kristina Denisova, Ph.D. was sole author. If you could learn every disease your child could possibly develop in life, would you? Newborn screening is required in the U.S. and differs slightly depending on which state you live in. For the most part, it's done before a newborn baby leaves the hospital and includes a blood test that screens for 30 to 50 serious health problems that usually arise in infancy or childhood, and could hinder normal development. Compared to current newborn screenings, genomic sequencing is able to detect many more inherited conditions. But there's also more to the results than just testing positive or negative, and with current commercially available products, those results and any caveats that exist may not be fully explained to the patient. Because of this, there's a push in the scientific community to determine how genomic sequencing should be implemented as a part of newborn screening before the technology is available on a large scale. Out of all the additional information we can provide through genomic sequencing, which could potentially be hundreds of conditions that might develop, what do parents want to learn, and what information would be most appropriate to provide, said Laura Milko, Ph.D., Associate Director of the Program for Precision Medicine in Health Care at the University of North Carolina School of Medicine. Milko is the first author of a study published in the Journal of Pediatrics that describes a method of sorting conditions to help categorize the types of results to be disclosed in newborn genomic sequencing. Milko and her colleagues identified several hundred conditions that could be helpful to report to parents. This study was part of a larger UNC School of Medicine project called North Carolina Newborn Exome Sequencing for Universal Screening (NC Nexus), led by Cynthia M. Powell, MD, and Jonathan S. Berg, MD, PhD. Current newborn screening standards were developed by the Health Resources Services Administration (HRSA) in their recommended uniform screening panel. Milko's study used this standard as a comparison tool in developing their sorting method. The conditions that are currently screened for in newborns are those that are more effectively treated if identified early, before symptoms develop, Milko said. We developed a sorting system that categorizes information in much the same way. Categorizing the information is important, because results from genomic sequencing include a lot of gray area, and some pieces of information are more helpful than others. We have around 20,000 genes and they all play roles in how our bodies grow, develop, and function. Changes in these genes, also called variants, might interfere with the gene's normal role and could lead to certain health conditions. Genomic sequencing is able to identify a person's genetic variants. Most of them are harmless or have no known health implications, but some variants might be related to rare genetic conditions. The variants that are pinpointed by genomic sequencing are an indication that a condition could develop, but do not necessarily identify when it will develop, or how severe the condition might be. Some diseases are more likely to develop than others, Milko said. We look at how many people within the population with that particular gene variant actually end up developing the disease to determine its prevalence. That information goes into determining how useful it might be to include in a newborn's genomic sequencing report. The group also looked at when conditions tend to develop. Current screening standards only recommend tests for conditions that develop during infancy or childhood and require immediate treatment. Genomic sequencing can uncover genetic variants that have an impact throughout a lifetime, but providing results related to conditions that could develop later in life has potential legal and ethical implications. Further, there is debate on how useful information is when no treatment is available for the condition that was identified. Would you want to know that your child has a chance of developing a potentially fatal disease at some point if there was nothing you could do to prevent or treat it? There are concerns that learning about a child's genetic risks might increase parental anxiety and have a negative impact on the family, especially when there is nothing that can be done about it, said Milko. There are also issues related to the disclosure of results about adult-onset conditions, since revealing this information about an infant would prevent them from being able to decide whether to learn it when they become an adult. Jonathan Berg, MD, PhD, Associate Professor in the Department of Genetics and Director of the Program for Precision Medicine in Health Care, was the senior author of this study. All of these factors played a role in how Milko and her research team sorted disease indicators into categories. They looked at 822 gene disease pairs altogether, and determined that 466 would be valuable to add to newborn screenings based on current HRSA standards. An additional 245 were identified as potentially useful, and suggested they be offered as additional testing if parents choose to do so. We need to start the conversation with physicians, policymakers and parents about how to use this technology, Berg said. The sequencing is really the easy part. The difficult part is thinking about what results to provide, how to teach physicians to share those results, and how to educate parents on what that information really means. The NC Nexus study is part of the National Institutes of Health's NIH Newborn Sequencing in Genomic Medicine and Public Health (NSIGHT) program. It was funded by the National Institute of Child Health and Human Development and the National Human Genome Research Institute. Simple test can tell if you're stressed out. Stress is often called the silent killer because of its stealthy and mysterious effects on everything from heart disease to mental health. Now researchers at the University of Cincinnati have developed a new test that can easily and simply measure common stress hormones using sweat, blood, urine or saliva. Eventually, they hope to turn their ideas into a simple device that patients can use at home to monitor their health. The results were published this month in the journal American Chemical Society Sensors. "I wanted something that's simple and easy to interpret," said Andrew Steckel, an Ohio eminent scholar and professor of electrical engineering in UC's College of Engineering and Applied Science. "This may not give you all the information, but it tells you whether you need a professional who can take over," Steckel said. UC researchers developed a device that uses ultraviolet light to measure stress hormones in a drop of blood, sweat, urine or saliva. These stress biomarkers are found in all of these fluids, albeit in different quantities, Steckel said. It measures not just one biomarker but multiple biomarkers. And it can be applied to different bodily fluids. "That's what's unique," he said. Steckel has been studying biosensors for years in his nanoelectronics laboratory. The latest journal article is part of a series of research papers his group has written on biosensors, including one that provides a review of methods for point-of-care diagnostics of stress biomarkers. Personal experience helping his father with a health crisis informed his research and opinion that a home test for various health concerns would be incredibly helpful. "I had to take him quite often to the lab or doctor to have tests done to adjust his medication. I thought it would be great if he could just do the tests himself to see if he was in trouble or just imagining things," Steckel said. "This doesn't replace laboratory tests, but it could tell patients more or less where they are." UC received grant funding for the project from the National Science Foundation and the U.S. Air Force Research Lab. Steckel said the military studies acute stress in its pilots and others who are pushing the edges of human performance. Pilots are placed under enormous stress during missions. The ground controller would like to know when the pilot is reaching the end of his or her ability to control the mission properly and pull them out before a catastrophic ending, Steckel said. But the UC device has widespread applications, Steckel said. His lab is pursuing the commercial possibilities. "You're not going to replace a full panel laboratory blood test. That's not the intent," Steckel said. "But if you're able to do the test at home because you're not feeling well and want to know where you stand, this will tell whether your condition has changed a little or a lot." UC graduate Prajakta Ray, the study's first author, said she was excited to work on such a pressing problem for her PhD studies. "Stress harms us in so many ways. And it sneaks up on you. You don't know how devastating a short or long duration of stress can be," Ray said. "So many physical ailments such as diabetes, high blood pressure and neurological or psychological disorders are attributed to stress the patient has gone through. That's what interested me." Ray said taking exams always gave her stress. "Understanding how stress affects you individually could be extremely valuable," she said. Stress has been a hot topic over the past couple years. Researchers have tried very hard to develop a test that is cheap and easy and effective and detect these hormones in low concentrations, she said. "This test has the potential to make a strong commercial device. It would be great to see the research go in that direction." UC is at the forefront of biosensor technology. Its labs are examining continuous sweat testing and point-of-care diagnostics for everything from traumatic brain injury to lead poisoning. Steckel, too, has been a preeminent innovator at UC. His papers have been cited more than 13,000 times, according to Google Scholar. In 2016, he used salmon sperm, a common byproduct of the fishing industry, to replace rare earth metals used in light-emitting diodes for a new kind of organic LED. "We're device engineers at heart," Steckel said. "We don't shy away from things we don't know much about to begin with. We look for opportunities. That's a hallmark of electrical engineers. We're not smart enough not to go where we shouldn't. Sometimes that pays off."